right, well, thank you very much for coming tonight. I'm going to talk a little bit tonight about plague. So, what is the plague? Most people, when they hear the word the plague, they think of the Black Death. So, the Black Death was a major pandemic that swept through Europe in the 1300s, killing 30 to 50% of all Europeans. So, it was one of those really traumatic major events in human history. And so to dive down a little bit into the details of what the plague actually is, it's a disease caused by a bacterium called Yersinia pestis. And so this organism, once it gets in your blood through a flea bite or other mechanisms, what it does is it typically moves to the lymph node, and it causes something called a bubo. So in the lower left-hand corner in bubonic plague, which I'm sure you've heard about, you can see this patient has a big swollen mass, and what that is it's bacteria that's traveled to the lymph node and is accumulated there. So the body's trying to fight it off. So in most cases, if you're in a developed country, you get antibiotics and you clear the infection. However, if it's untreated, it can move to the pneumonic form. So the pneumonic form, it moves to your lungs, and then you can cause person-to-person transmission of plague. And this is where, if you're not treated immediately, the chance of death is about 100%. So it's a really fatal disease if it's not treated in an effective way. So what many people might not realize is the plague is still present throughout the world today. So this is a map with the countries in red showing where we know plague to currently exist. So that includes the United States, but also South America, Africa, Asia, and there's countries that aren't shown because maybe they're not reporting that they have plague. So this is where we know it to exist, but it's not comprehensive, so it might be much more extensive. And so, again, when you think of the plague, you might think of the Black Death. I think of something more like this. So the black-tailed prairie dog here in northern Arizona is an organism that will get bit by an infected flea. So when it gets bit by an infected flea, it gets the plague, it crawls into its burrow, and it dies. And so the flea hops off, and it waits for the next prairie dog to come by. So this is really the major reservoir where we see plague here in northern Arizona. So the map in the lower right shows really where plague is. So it's a disease in the western United States. And a lot of this has to do with the species of not only fleas, but as well as rodents that carry those fleas. And so you can see, it's in Flagstaff, it's all over northern Arizona, it's in New Mexico as well as southern Colorado. So it's something that we have to think about living here, and it's something we have to consider. And it's not something that's, a huge risk for living here, but, this was a couple years ago where there were about 4 deaths and 15 cases of bubonic plague in 1 year. And so this is why, one of the reasons why I really wanted to study this organism is, it has these huge historical implications, right? So it was involved with the Black Death, yet you can go outside, and sample for it, in our backyard. So it's a disease that has a lot of, historical as well as local implications. And so as biologists, one of the tools that we use to understand the relationship between living things is something called gene trees. And so this is a model that we can understand how things are related by using different lines of evidence. And so as a geneticist, we use a lot of DNA sequence data. And so this tree that you're looking at here is a relationship between primates. So you have the greater apes, humans, and chimpanzees. You have the lower primates and monkeys. And what you can do is you can compare the DNA between those organisms, and you can build this model to try to understand how things are related based on how shared that DNA is to one another. And so in the case of humans and chimpanzees, humans didn't evolve from chimpanzees, but we share a common ancestor. And this is important when we're trying to infer these relationships. And so with humans and other primates, the nice thing is we can go and we can obtain fossil evidence, or we can look at skeletons or skulls. And that information can also inform our gene trees. In the case of bacteria, we're just limited to the DNA sequence data. So this is a tree looking at plague. And so each one of these little tips, a circle, a triangle, a star, represents a plague isolate from a different location across the world. So at the root of the tree, just like the relationship between humans and chimpanzees, we see a common ancestor. And that's the common ancestor between plague and really the closest relative to what plague has. And so the way to interpret this tree is as we go up the tree, we can think about it as time increases. So we're getting further and further away from that branch with a common ancestor. And as it develops these different branches is where plague is, located to a certain area and then established there. So I circled a figure of China. So that clade with the little green circles might indicate a group of plague that has colonized China and is kind of localized there. So if you wanted to go sampling for it, you could go to a certain location in China and find that exact lineage of plague. And so this kind of, over time, you can see there's kind of sporadic establishment of plague in different locations. But then when you get to this location on the tree, we see something very different. And so we call this the Big Bang polytomy. And this is where plague really went worldwide. And so the reason why we think this is because humans started going worldwide. People got on steamships, they traveled to North America. Well, within those steamships, we had rats, and rats had fleas. And so you think as you land in the new world, the rats get off, the fleas get off, and all of a sudden you have a new location that can establish. And so this is really where plague established across the world, including Arizona. So if we look at this, where Arizona plague lies, it's right here on the tree. And so we know a lot about modern plague because we can go out and sample for it. We can either sample for it here in Flagstaff, we can work with collaborators across the world to look at their plague. But there's only so much that plague can tell us. If we want to look at some of those ancient incidents of plague, like the Black Death, we really need to look at ancient DNA. And so ancient DNA is a technology that has really only been established at a really high level in the last number of years. And in terms of looking at it in terms of infectious disease, if we wanted to look for plague or other infectious diseases, the best place that we figured out to look for it is in human teeth. So human teeth, you have the dental pulp. It turns out when you die and you're buried, it's kind of a time capsule that preserves the DNA. It's still broken down, but it turns out to be intact enough that we're able to work with it. So when we obtain a tooth like this, what we can do is extract all of the DNA in the teeth. And so what you're mostly going to get is human DNA, represented by red here. However, you can also get, there's going to be a small percentage of the DNA that's going to be bacterial in origin, represented here by orange. And the fragmented nature of these little cartoons demonstrate that the DNA is not going to be totally intact. It's going to be broken up because it's been degraded over time. So we have technologies now that we can then enrich for the bacterial DNA. So we're a little bacterial focused in our laboratory. We don't care about the human stuff. So we can enrich for the bacterial DNA. And then we can put it on a gene sequencer. So here in Flagstaff, we have several of these gene sequencers. And what you get at the back end is the genetic code of that organism. So for ancient DNA, we get this genetic code. We can compare it then to the genetic code of all the modern day plague isolates that we have. And we can start using those gene trees as a model to compare and see how they're related, see how plague has moved across the world over time, and give us more insight about exactly the nature of some of these large pandemics that we know to be so important in the evolution of people and human society. And so I'll talk to you about several different stories today. The first one is Bronze Age plague. And so the Bronze Age was thousands of years ago, and researchers started unearthing these graves that they knew were from the Bronze Age. They could date these skeletons based off several lines of evidence. And they knew they were very, very old. And one of the questions they were asking is, why did humans migrate so extensively during this period? They knew there was these mass migrations throughout Europe. And so the researchers were wondering, well, was there some big infectious disease incident where they had to flee? Maybe there was too much inbreeding within a population. They had to branch out. So the researchers were really interested in, can we, from the DNA, determine why these migrations happen? Well, within their studies, they unearthed 101 skeletons. So 7 of these skeletons ended up being positive for something that was plague-like. So we could run it through our pipeline, we could look at the genetic code, and we could place it into that tree model to see, well, how is it related to the plague that we know to exist today? So kind of not surprisingly, the Bronze Age is in between all the plague that we can see today and the nearest ancestor, which is about, from 52,000 BC. So to some extent, this wasn't that surprising. But as we start looking at the DNA, we can start to answer other questions as well. So just looking at the DNA, we believe that the plague was not capable of colonizing fleas. So the transmission cycle that I discussed earlier is not something that we think this Bronze Age plague could do. So how did it infect people? And the answer is we're not sure yet. We're hoping to answer that with more lines of evidence and improved technology. And so that was, 2000 BC. And if we look at the history and we try to figure out what other pandemics might be associated with plague, the next thing that we really think that was associated with the plague was the plague of Justinian. So if you haven't heard about the plague of Justinian, it happened in about 540 AD. And it was, again, a really, really traumatic event in human history. It ended up killing between an estimated 25 million to 100 million people. And so if you think about the world population at that time, that'd be like killing billions of people in today's world. So it was a really, really major event. It helped, bring down the Roman Empire. There was too many deaths. The Roman Empire couldn't subsist anymore. So it was one of the events that helped bring that about. And so we had collaborators working in Germany. And as part of a housing development, they excavated some land, and what they found underneath was this mass grave. And so when we find mass graves, it's usually indicative of a huge, traumatic event, killing lots of people where they just buried them all at one time. And so as we started dating these skeletons in the mass grave with both, artifacts that were nearby as well as carbon dating, we realized that these were from the plague of Justinian. And so we looked at the teeth were positive for plague, and so we could then put that into that tree model to see, okay, well, where did this, plague of Justinian come from? And so if we look at this tree, what's interesting about the way we interpret it, it looks like there was a lineage of plague that caused this plague of Justinian, and then as far as we can tell, it died out. So it emerged, it killed all of these people, and then we do not see modern representatives of the Justinian plague. So that was kind of an interesting finding. We expected, well, maybe this Justinian plague turned into the Black Death. Doesn't seem to be the case based on the analysis. And so now the plague of Justinian happens. The next really recorded incident of plague is the Black Death. So I already mentioned this. This was a major event in human history, killing 30 to 50% of all Europeans. So this was a mass grave that some collaborators unearthed outside of London. So they looked at the teeth, and we sequenced the DNA. When we look at the placement of this on the tree, it's a much different interpretation. So we believe that the Black Death is a direct descendant of really the plague that went worldwide, went to North America, the same plague that we find here in Arizona. So this is a direct descendant. And so what that means is that the plague that's in Flagstaff has all the genetic machinery to cause that huge pandemic, to kill all these people. But we have antibiotics, and we have, germ theory, and we have hygiene, and so we have ways to combat this. But it's interesting to think that the plague that's in the prairie dogs is a direct descendant of the plague that caused the Black Death. And so after the Black Death, we don't hear a lot about plague in the history until there was an outbreak in Marseille, France in 1722. And so this was another big outbreak. One question that we had when we were looking at these samples, we started to get the DNA sequence back, is this the same Black Death organism that's just been circulating in Europe for hundreds of years? Or maybe was it a new introduction, a reintroduction of plague back into the area? So we looked again at these skeletons, looked at the teeth, and when we look at the placement on the gene tree, we now believe that this outbreak in Marseille was a direct descendant of the Black Death. So we do think that it was circulating in rodent reservoirs for hundreds of years, and then it died out. So this is really the last gasp of plague in Europe, and it's been eradicated ever since. So through these types of tools, we can start to understand exactly how they persisted, was it reintroduced, and exactly how it was transmitted. So DNA applications, ancient DNA holds a lot of promise for answering lots of questions about how organisms emerge, how they spread, and really how they've impacted human history. And so I've talked about a few examples today. We have to remember this technology is really in its infancy. So now that we know that these types of analyses are capable, we're going to start looking at more skeletons. We're going to look at, with better technology, and we'll hopefully be able to fill in some of these question marks because, between the Bronze Age and the Justinian plague, we don't really know where plague went. We don't know how it changed and became really the nasty pathogen that it is today. And so these ancient DNA applications are things that we're starting to take to other infectious diseases. So we can look at mummies. So this is a mummy that was infected with tuberculosis. And so tuberculosis is a disease that we know still exists today. So we can compare the tuberculosis of thousands of years ago to see how it's changed and if it's become more infectious or not. We can also look at infant mummies. And so infant mummies, this is an infant mummy who died of smallpox. And so by going in, looking at DNA, we can start to reconstruct what that smallpox organism looked like, and then understand a little bit more about transmission and what it looks like compared to, cases that happened within the last couple hundred years. So ancient DNA really holds a lot of promise to understanding more about the nature of infectious diseases from a historical context. So I work, again, in the biology department at the Pathogen and Microbiome Institute. So we've really assembled a kind of a world-renowned team to look at this DNA type of sequence analysis, but we do lots of other things. So I encourage you, if you're interested in what we do, to want to see what other types of studies we do, I gave you the web link here, and we're always looking for talented undergraduate and graduate students who want to do DNA-based research on infectious diseases. Thank you.